Thank you so much for joining us today in this webinar. Thank you for waiting. We have an excellent speaker today, Hurui Kui. I will go with the introduction. Hurui Kui is a senior scientist in the drug discovery function at Foghorn Therapeutics. Hurui earned her PhD in chemistry from the University of Minnesota Twin Cities, where she worked in the lab of Professor William Pomerantz. During her doctoral research, she developed one of the first and still among the most potent selective BRD4 D1 inhibitors, contributing important early advances to the field of selective bromodomain inhibitors. Following her PhD, Huarui completed a postdoctoral work with Professor Angela Kohler at MIT, where she gained deep expertise in high-throughput screening and cell-based assays. Hurui joined Foghorn Therapeutics in 2022, where she now focuses on designing and executing high-throughput cellular screening strategies, validating hits, developing target engagement assays, and driving mechanistic studies to support early-stage drug discovery programs. From here, Hurui, it's all yours. Take it away.
Thank you, Amar. Thanks for the really nice introduction, and thank you for having me here today. So today, I will tell you a little bit about how we use the microtag technology to assess cellular target engagement at Foghorn Therapeutics with ARID1B as a case study. And also, I will highlight some progress we've made on the selective ARID1B degrader. And if we go to the next slide, so first, I do want to emphasize why cellular target engagement is important for drug discovery. So first of all, cellular target engagement allows us to understand whether your small molecule of interest is truly engage your protein target in cells under a more physiological relevant conditions. And when we run biophysical biochemical assays with recombinant proteins, the proteins, they're under a more isolated environment. And it may not truly reflect how it works in cells because those proteins might be in a protein complex when they're in cells, or they might have different conformational change. So we do believe cellular target engagement assays could provide us more information. Also in the literature, there are more and more evidence showing that the proteins, especially those ones with the intrinsic disorder regions, they tend to have some transient structure in cells, and those transient structures cannot be captured by biophysical or biochemical assays either. So that's why we think with cellular target engagement assays, it can provide us a more accurate representation for how protein works in cells. And we do believe those information can help us a lot in drug discovery as well as supporting support and drive SAR study for the lead optimization. And if you go to the next slide, so at Foghorn, we've developed a whole bunch of cellular target engagement assays to learn our compound activities. So here I'm showing you a few examples. So the first one I showed here is the nanobright assay. So nanobright uses energy transfer to measure the small molecule and protein interaction. And as you can see here on the left of my slides, so it requires two elements for this energy transfer. So first, you need to have a nanolook tag on your target protein, and then you need to have a small molecule tracer that can bind to your target protein. And when the small molecule binds to your target protein, it will generate a bright signal. And then you can treat with your compound of interest to compete off the fluorescence tracer. And this experiment can give you a quantitative as well as a computation-based binding affinity. Sorry, could you go back to the slide? Thanks. And although the nanobright is super high throughput, it has its own limitation. For example, this a specific experiment requires the tagging system. So you have to have a nanolog tag on your target protein, and also it requires high quality probe. So the probe needs to be cell permeable for this experiment. And also this experiment can only give you on-target effect, but not off-target effect. We also enabled nanobread ternary complex formation assay at Foghorn. So for this experiment, it's extremely helpful to provide information about the ternary complex formation induced by the bifunctional molecule as well as molecular glue. It's also a really high throughput assay, and also it only detects on-target effect. Also, it requires two tagging protein. One was halo tag and one was nanolog tag. But it can give you tons of super important information for induced ternary complex formation. And we also enabled other experiments for cellular target engagement. So here on the right, on the left, I'm showing you the affinity-based protein profiling for covalent binders. So here I'm using the cysteine direct ABPP as an example. So in the workflow, first, you need to treat your covalent small molecule in cells. And then after the incubation, cell lysase, you need to incubate the cell lysate with a cysteine reactive probe. And then the enrichment will happen and mass spec will be used to determine the degree of computation between your covalent binder and the cysteine reactive probe. Even though this assay is slightly lower throughput, it gives you tons of important information about the specific binding site of your covalent binders, as well as all the off-target information. However, for the detection, you have to have a tryptic peptide to be detected by mass spec, and also the cysteine of interest needs to be reactive with the cysteine reactive probe. Could you go back to the slide? Thanks. And we also enabled photoaffinity labeling assay as well for the non-covalent binders. So for this specific experiment, first you need to add this photoaffinity enriched alkyne handle on your molecule of interest, which is showing in blue and red on my slides on the right. And after the incubation, UV irradiation will be used to active this diazo ring, and it will covalently cross-link to all the nearby proteins. After enrichment, all the proteins that are being labeled by the molecule of interest will be pulled down and then detected by mass spec. So again, this assay is still relatively low throughput, but it will tell you the on-target as well as the off-target information. So this specific assay usually suffers from low labeling efficiency and some non-specific labeling, but it can give you super important information about the off-targets. And at Foghorn, we are also interested in other alternative target engagement assays, including the microtag technology. And in cells, there are thousands of proteins, each with distinct structure and stability properties. So when intact cells are exposed to temperature gradient, a soluble protein can go through a thermodynamic change, a temperature-dependent denaturation, and aggregation. So a fully soluble protein could transient from fully aggregate protein. And through this whole process, there are tons of intermediate that might be reflective for the different conformation of the protein adopted in cells. And for microtag technology, it can capture and monitor the cellular protein stability across the temperature series and measure the shift induced by the small molecule. And if we go to the next slide, this is how this experiment works. So to start with, you need to label your protein target with a small tag, a 15 amino acid tag, which is the S tag I showed on my slides in purple. And this could be done either by endogenously knocking or overexpression. So after you have the reporter system, then you can treat your compound of interest in cells. And if the compound is capable to bind to your protein of interest, then it should induce a thermal shift after applying the temperature challenge. And then for the detection, you could add the S protein, which is the one I showed in orange here, as well as the RNA fresh substrate. So the S protein is the larger subunit of the RNA enzyme, and after it recognizes the S tag, they will reconstitute the active RNA enzyme, and then the enzyme can cleave the RNA fresh substrate and then release the fluorescence signal. So the fluorescence signal, the magnitude of the fluorescence signal is directly proportional to the soluble protein in cells. And the detection system could either be a regular plate reader for end-time point readout, or you could use the qPCR machine for real-time readout. And this is a pretty straightforward system. It's just mix and detect reaction system. And you could do this in either transient overexpression or stable expression of the S-type protein. And also it's fully programmed in the qPCR machine. So it's really easy to set up. And also this specific technology doesn't really need to have a probe to compete off, or you don't have to do any modification on your small molecule. But you do have to have the S tag on your protein of interest. And here is how the data look like. So because we are applying different temperature challenge, so you can see at different temperature, the test compound can either stabilize or destabilize the protein compared to the DMSO control. And then we apply the linear regression analysis for each temperature. And on the right, you can see the heel slope of the tested compound as well as the DMSO control. Then we can plot the linear regression of the heel slope against different temperature or protein concentration or small molecule concentration, sorry. And then it will give us the engagement potency, the EC50 here. And at Foghorn, we are interested in the chromatin regulatory system. And the key point here is that the structure of the chromatin dictates its own function. So for open chromatin, it leads to gene expression, and for closed chromatin, it represses gene expression. And the proteins we are interested in actually control the gene expression by changing the chromatin structure. And one of the proteins we're super interested in is the ARID1B I showed on the slides. And if we go to the next slides, so we're trying our approach to address the large population of ARID1A mutation is to selective degrade ARID1B. And it leverages the synthetic lethal relationship between ARID1A and ARID1B. So as I showed on the left of the slides, you can see in the tumor cells, they're dependent on the ARID1B for survival. However, in the normal cells, because they have the intact copy of ARID1A to compensate ARID1B degradation, then the ARID1B degradation is not as important. And this is also supported by our colony formation data I showed on the right. So the selective ARID1B knockout only affected the proliferation of the ARID1A non-cell lines. However, ARID1B is a really challenging target to drug with few known binders. So first of all, it's a really large protein with a lot of highly unstructured region. So the molecular weight of ARID1B is over 240 kilodalton. And as I showed here on the sides, it has a lot of disordered region on the specific protein. And it has no known enzyme function, which makes it really difficult to develop biochemical assays. And also, it's a subunit of a large multi-subunit protein complex. And as I showed here on the right, this is a cryo-EM structure of the nucleosome bound buff subunit. And there are tons of other subunits around ARID. And also, it has really high sequence similarity and also structure similarity to ARID1A. With all of this, targeting ARID1B is actually super challenging. However, at Foghorn, our platform did yield several selective ARID1B binders. And here on the left, I'm showing you the plot from primary screening results. And as you can see here, I'm trying to highlight numerous of selective ARID1B degrader over ARID1A. And on the right, I'm trying to show you some ITC traces from one of our binder. It showed strong selectivity against ARID1B over ARID1A. Yep. And so far, to date, we have made substantial progress on improving the binding affinity of our internal binder through the structure-based optimization. And as you can see here on the very left, we started with a binder with around 100 micromolar binding affinity. And after rounds and rounds of optimization, right now we have a lot of binders with sub-micromolar binding affinity. And when we're doing the optimization, we also take other parameters such as solubility, media stability, and also selectivity into consideration. And with all the binders in hand, we were also wondering if the microtag technology could help us to assess the cellular target engagement with those binders. And to take the initial step, the team at CellarisBio, they were helping us to overexpress the S-tagged ARID1B in 293T. And as you can see on the top left corner, there is a band corresponding to the expected molecular weight for ARID1B on the Western blot. And then after adding the S-tag binding protein and the FRET substrate, you can see a really nice fluorescence signal there. And then we also assessed the aggregation temperature after we have this reporter system. So as you can see here, with increasing the temperature, the soluble fraction of ARID1B decreased, leading to the reduction of the slope on this temperature gradient plot. And then we plot the slope against different temperature, which give us the TAC50 for ARID, which is around 48 degrees Celsius. And after we see the results, we evaluated a handful of our internal compounds with this technology. And here on the left, I'm showing you one representative example. And as you can see here, our compounds show the clear separation across different concentrations, especially from the top or the highest concentration to the lowest concentration. So the highest concentration is shown in black and the lowest one is the one in purple. And then we applied again linear regression on the hill slope at 50 degrees Celsius, and then we plot that hill slope against concentration of our binder. And as you can see on the bottom left, we can quantify the EC50 from this temperature plot. And the molecule we're using in the microtech technology I showed here is also a PAL probe. So we also test the specific molecule in the PAL assay. And as you can see in the bottom right, with the UV irradiation, you can clearly see ARID1B being pulled down from the cells, which is also really encouraging to see both assay showed target engagement in cellular context. However, the EC50 we got from the microtech assay is slightly higher than the biophysical KD value we got, but this discrepancy seems pretty common. If you go to the next slide, yep, the team at CellarisBio, they also tested other reported small molecules. For example, I'm showing two here, one for MAPK1 and the other one for KRAS. As you can see here, both molecules actually had really potent IC50 in the biochemical assay. However, their EC50 in the cellular experiments tend to be slightly higher. And we do think this could be due to the fact that the cellular target engagement is capturing the engagement in a more physiological relevant condition. And also there are a lot of factors that could affect this EC50, such as the permeability of the compound. And in parallel, we also advanced our selective ARID1B binder into ARID1B degrader. And actually, so far, we have progressed a lot of degraders with over 80% ARID1B degradation with both cereblon and VHL handle. And here I'm just showing you one example from our degrader series. And as you can see here on the left, I'm showing you the immunoblot for the degrader. And across different concentrations, we see really nice and strong ARID1B degradation. And we also showed you the endpoint habit readout for the ARID1B degradation in a different cell line. And also we used the kinetic to evaluate our degrader as well. You can see a really nice ARID1B degradation was in a really short time point, and this is super encouraging. And as you can see here, we also try to confirm if this ARID1B degradation is on mechanism effect via the ubiquitin proteasome system. And on the left, I'm showing you that when we incorporate the VHL dead ligand or the inactive ligand for VHL, we don't see any ARID1B degradation anymore. The degradation is only with the active degrader. And on the far right, I'm showing you that our degradation can be rescued by different proteasome inhibitors such as MG132, bortezomib, and also methylation inhibitor MLN compound, and also E1 inhibitor, the TAC243. And on top of that, we also showed that our compound didn't really show any cellular toxicity at 24 hours across the concentrations we checked. And this further gave us confidence that the ARID1B degradation is on target mechanism, not through the off-target proliferation. And I didn't show the data here, but we do have a global proteomics data to show that our degrader only selectively degrade ARID1B across the whole proteasome, which is also really encouraging to see. And with that, I hope I showed you how we use our internal program ARID1B as the case study to evaluate microtech technology. And also, it's really encouraging to see that our selective ARID1B binder showed target engagement in cellular context. And also, we will continue evaluate cellular target engagement assays such as microtech technology. And also, I hoped I showed you the progress we've made on selective ARID1B degraders. And if you go to the next slide, and with that, I would like to thank everyone at Foghorn Therapeutics for their support. And also, I would like to thank everyone on the team for their efforts to enable this work. And with that, I'm happy to take any questions.
Please leave your questions in the chat box and we will try to answer them the way they arrive. One question, Rui, for you. So you mentioned that you are using there are two ways to use the data for data analysis of the microtag data. This is the area under the curve and the slope regression analysis. But how do you actually program the machine? So
Yeah.
in real-time qPCR systems, what exactly do you do to program the microtag experiment?
Yes, so I think we have a backup slide for the programming in the deck as well. If you could.
I'm not sure we have a slide for that. Maybe if you can just talk to that.
Yeah, so the whole programming is actually pretty straightforward. And so basically how it works is that you could set up the temperature to you could set up like the temperature gradient you want to do, and then for each temperature, it will stay ramp to the temperature, stay or incubate at that temperature for a specific time, usually 3 minutes, and then it will go back to 20 degrees Celsius, do the read, and then ramp to the second temperature, do the same thing back to 20 degrees Celsius to read, and then eventually it reach out to the temperature you are interested in. So the one we did is from 30 or 25 to 70 degrees Celsius.
Thank you for that.
Yep.
Another question that has arrived from an anonymous attendee is, do you use the same critical path for various challenging targets in your drug discovery programs?
Um, could you specify the question a little
I
bit more?
guess what
I
they
didn't.
mean is that you have shown a specific series of experiments you used for ARID1B study. Do you use identical series of experiments or do you shift around and develop a new critical path?
Yeah, that's a good question. I guess it depends on the protein targets and also how you want to screen the protein. So for different proteins, we do have different hit triage, the way we screen, they're all different. So the assays are designed based on the property of the protein itself. So we don't really have a specific workflow for all the proteins, but we design new experiment for specific proteins.
Thank you. Another question, do you need to optimize the N-terminal or C-terminal linked S tag in the microtag experiment?
That's also a really good question. Yes, we do. And usually we try both to start with and see which one works. And for this specific experiment, we know the C term would work for us. That's why we started with the C term tag.
Another question is maybe it's a repetition of one of the previous ones. Does thermal cycling compromise the cellular context? Wouldn't the cells degrade after a cycle or two?
So uh, I guess yes, it depends on the temperature you're using, right? So if you go to 70 degrees Celsius, for sure the cells won't survive under that high temperature. But if you go to, let's say, probably slightly higher than 37 degrees Celsius, but not too high, the cells might be okay for a specific time of point, for a specific time. And also based on our experience with the CellarisBio, we do see that at least within 40, 50 degrees Celsius, the protein itself is still there. You can still see like the fluorescence signal after you add the Aztec substrate or Aztec binding protein and also the fresh substrate. And we are not like for the specific assay, I guess we're more interested in whether the protein can still hold the structure, not the cells.
Right. Um, Thanks for the nice call, Huarui, for nice talk, Huarui. Could you please elaborate on how microtag compares with other methods such as SETSA and hybrid tag based BITSA for cellular target engagement?
Yeah, that's also a really good question. I think for SETA, uh, one of the advantage of SETA over the other, let's say the beta and the microtag technology is that you don't have to have the tag on your protein of interest. You could just use Western blot as the detection. However, that's also the disadvantage because you have to run Western blot all the time. And for microtech technology or beta, you could just use either luminescence or fluorescence as the readout. So there will be more higher throughput. Um, I guess that's my answer for the specific question. The other one I know about beta is that I think you have to do endpoint readout for beta because you cannot really do a real-time readout because it's a luminescence readout. But for microtech technology, you could read at each cycle during the experiments. So that could give you some additional information.
Thank you. Next one is, how is the S protein delivered into the cells for complementation with the tag? Is the protein added during or after the temperature challenge?
That's also a really good question. So the S protein is added after the incubation with the compound before the temperature challenge. And the S protein is always there during the whole experiment.
That's
And
correct.
I think the CellarisBio team also added tritin to get the cells more permeable for the S protein to get into the cells.
Right. It's important to note here that S protein is actually thermally very stable. So as we increase through multiple temperatures, S protein will stay stable. It will lose some activity at very high temperature points above 75, but otherwise it's very stable.
Mhm. And
Next
also, that's
question.
a really good point because the reason we detected the aggregation temperature of ARID1B is to make sure, like the melting temperature or the aggregation temperature of ARID1B is not too high. So when we run the experiment, the S tag protein, it's still active.
Okay, thank you. Questions keep coming, so we do have some time though. Microtag is good for challenging drug targets. What kind of drug targets might microtag not be suitable for? Can
Mm.
you answer that, Rory?
That's a really interesting question. I guess we have to try and figure it out. But I do think so. Well, Aztec is actually really small. It's only 15 amino acids, so it should not really affect the thermal stability of most of the protein. But if you do have a protein that's super stable, like what we mentioned earlier, then this technology may not be suitable because the S protein as a whole may not be stable anymore.
Right, so empirically needs to be tested, I guess.
Yeah.
Every target is unique, and therefore we just empirically need to test to elicit that response. Next question is, why did you choose the slope of a fluorescence change in response to the temperature challenge instead of endpoint fluorescence at the end of the cycling at each temperature?
That's an interesting question as well. I think for beta, they do use the endpoint readout for the setup. And we do think so for microtechnology, one of the advantages is that they can read the fluorescence signal at each different temperature. So we do think that might be helpful for us to get additional information. So that's why instead of doing endpoint readout, we decided to do this live reading.
Next one is how stable is the S tag itself, and how is the technology suitable for membrane proteins?
Mm, I
Is this
think
something you can comment on?
this might be a question for the CellarisBio team because that we don't work with the membrane protein.
Right, maybe I can comment on this. So, S tag itself is a soluble tag. It's not hydrophobic and therefore can work well with membrane proteins. Every target, again, is very unique, but we do have cases with multiple membrane proteins. Challenge with membrane proteins is that they do not completely melt, so the thermal profile will never come down to the baseline. But we can work with membrane proteins, but then again, we have to empirically test and see what the response, the unique fingerprint looks like.
Mhm.
Another question, Horui. Promega recently reports Brett's target engagement technology. Do you have any thoughts on that?
So we are actually the beta tester for the Beta technology as well. So we will keep you posted on what we think on the Beta.
All right. Is ARID1B a monomer or a complex with other proteins?
Yeah, so ARID1B is in buff complex. It's a gigantic protein complex with tons of other subunits, and that's why mm, it's also a really challenging protein target because you always work with other proteins around.
If both N and C termini are challenging for adding the S tag, is there another way to add the S tag to the target protein?
I think this might also be a good question for you, Omar.
Right. So we do prefer N or the C terminus, one or the other. And if there's biology related to on one end, we prefer the other end. And again, we by default we prefer the C terminus. We do not use S tag internally, not currently, but maybe in the future we might. So if there are sensitive functions on both N and the C terminus, we just have to make a trade-off and pick one of them. Currently, that's the state of the situation with us. Is the strength of this technology is its ability to elucidate the mechanism of action when one already possesses a compound capable of degrading a specific protein, or is its strength rather the ability to identify a suitable degrader when one is interested in a specific protein, or is it both?
Do you want me to take
I
on
mean,
this?
you can try.
Yeah, mm, that's a good question as well. I guess it depends on what you want to learn from the specific technology. So for me, I think this is a pretty interesting essay to learn cellular target engagement for binders instead of degraders. But I know for CellarisBio, they also have new platform about to like detect protein targets. And I think Omar might be able to comment more on those.
That's correct. So we can be agnostic to whether there is already an existing inhibitor to a target or not. This technology can be used for discovery, de novo discovery of the chemical matter, as well as for validation of candidates after the fact of discovery. When it comes to discovery, I mean, we can use various approaches here. We can use, for example, cellular DEL discovery. We can use this technology to bring in DEL libraries and discover the chemical matter. or we can use it for conventional drug screens, plated drug screens. But one of the conventional uses of microtag is when you already have a drug candidates in hand, because then it's validation. And then the question is, do we have positive controls or not? If yes, that's great. If we do not have positive controls, then we might want to work with some general promiscuous compounds. There are various situations we can assess based on the target and based on the discovery program. Maybe we can take one more question here. Um, does Cellaris have plasmids in which the target of interest can be cloned into an add S tag onto the N or the C terminal? Yes, we can offer our reagents for beta testers. And in fact, we're running this open target initiative currently. So if there is interest, please write to us. We will respond. One more question for you, Huarui. What specifically makes some drug targets challenging, and how does microtech specifically address those?
Yeah, I guess a lot of things could make some specific targets challenging. For example, like for transcription factors, they don't have the defined binding pocket, which make it really difficult for a small molecule to bind to it. And for ARID, for example, it's a gigantic protein. It's in a big protein complex. And also, it has so many intrinsic disorder region. So all of those could make the protein a super challenging target. And I guess, as I mentioned early in the talk, cellular target engagement assays, they could give us additional information than some biochemical, biophysical assays because your protein is under a more physiologically relevant condition.
That's right. So I guess we can take last two questions. They're short ones.
Mhm.
Does the S tag affect the accessibility of the probe for ARID1B?
So first of all, for microtag, it doesn't really need a probe. And secondly, for the S tag itself, because it's only 15 amino acid, so we don't think it should affect the binding of the small molecule to the large ARI1B protein.
I guess the last question is for CellarisBio team. Can microtag be used for cellular DEL, I guess, DNA encoded libraries? The answer is yes. This is one of the new applications we're developing, and we will have a webinar on this soon. But yes, a microtag enables de novo discovery of chemical matter from DNA encoded libraries. And in fact, this is very much useful for challenging targets that cannot be used in DEL discovery using the conventional methods. How do I subscribe for future webinars? Just write us an email and we'll put you on the list. Thank you so much, Huarui. Thank you to all attendees for listening to this webinar. We will see you soon, and please stay in touch and please subscribe for future events. Thank you so much. See you soon.
Bye.
Bye.